Hello, my name is Evelyn Yim. I'm Associate Professor at the Department of Chemical Engineering in the University of Waterloo. Today I have the opportunity to share my work on the sensing biomaterial topography through Mechanotransduction in engineering cell niche. So first of all, I would like to thank the organizer for giving us the opportunity to share our work with you. Our lab is interested in the research of tissue engineering and regenerative medicine. The concept of tissue engineering has been proposed in the 1990s, in which the motivation is to generate or engineering functional tissue or functional cells to repair or replace damaged tissue in the patient. The concept involves isolating cells or stem cells from a patient, and then we will culture them in vitro, generating functional tissues to replace the damaged tissue. Another branch of regenerative medicine involves having cells from a patient or from a donor, and then we will manipulate or regulate the cell fate or cell function and implant these functional cells back to the patient as cell therapy. So in both methods, one of the important steps is to understand how to regulate stem cells, the stem cell fate or the cell's behavior in vitro. So as we know that we can regulate cells' behavior by using chemical like growth factor, cytokine, extracellular matrix protein, or even small molecule drug. And research in the last decades also show us that mechanical factors such as the rigidity of material or by controlling the geometry of cells could also affect cells' behavior. Our lab is particularly interested in the topography of the extracellular matrix, and we, as well as other groups, have shown that this substrate topography, when we replicate on synthetic material, this substrate topography could affect different cell response. And in fact, this type of extracellular topography can find in many types of tissue. These two pictures are two electron micrograph picture find from the internet, but it shows a very rich reservoir of extracellular topography found in the vasculature. Another electron micrograph picture shows here is the topography in the corneal epithelial basement membrane. So this evidence shows that cells are surrounded by a rich reservoir of extracellular topography. Our lab is interested in applying this type of topography for tissue engineering application. So how can we apply this topography for? We have been working on different types of application. So we have been interested in applying the topography on neuronal differentiation, vascular application or vascular repair, as well as corneal regeneration. For today's talk, I will be focusing on talking about how does it work or why does it work. Our group has been looking into the mechanobiology in the topography-induced cells behavior, including topography-induced stem cell differentiation. We find that topography-induced differentiation is dependent on the geometry of the pattern. An important component of the topography sensing is the actin-myosin's contractility and the focal adhesion signal. So before I start talking about the data today, I would like to acknowledge the contributions from my research team as well as our collaborator. So I highlight the lab member or my student and postdoc who has contributed directly to the presentation I'm going to give today, and also show some other of my lab members both at the University of Waterloo and also in the National University of Singapore in Singapore. So I have been a principal investigator and a faculty in the National University of Singapore before I relocate to the University of Waterloo in 2016. So some of the work I show today are also generated from my lab member, my former lab member from Singapore as well. In addition to my lab members, I would also like to thanks and acknowledge my helpful collaborators as well as the funding source that enable us to do the work. we start investigating the effect of topography on stem cells differentiation on the effect on the neuronal differentiation. So in this study, we cultured human embryonic stem cell H1 and H9 on different type of topography. In general, we find that pillar substrate we usually will favor and enhance astrocyte differentiation, versus grating substrate will prefer or favor the neuronal differentiation, and they will also enhance neuronal maturation. The period of time required for the neuronal differentiation is usually shorter than compared to unpatterned substrate or other protocol using unpatterned substrate. We have also investigated how to derive neuronal subtypes using a combination of topography with other protocol. My former postdoc, Kenneth Tan, has taken this work to differentiate induced pluripotent stem cells through a two-stage protocol, and then he incorporated topography in stage 1, which is the lineage commitment stage, as well as stage 2, which is the maturation stage. We find that while in stage 1 in lineage commitments that gratings do enhance the neuronal differentiation, in stage 2 in the maturation stage, both gratings and pillars will also help to enhance the maturation and derivation of neurons. To investigate or to examine the derivation of dopaminergic neurons, we examine the expression of tyrosine hydrogenase, which is a marker of dopaminergic neurons. On grating, we observe that there's a significantly higher percentage of tyrosine hydrogenase positive cells on gratings. We also have higher TH positive cells on pillars compared to unpatterned as well, although it's not as significant. Not only that, when we examine the electrophysiology or the electrophysiological functions of the neurons, we find that there are more neurons capable of firing repetitive action potentials on the patterns, on both the gratings as well as the pillars pattern. In our recent work, we have shown that using topography patterns, it can also enhance the derivation of induced neurons by using non-viral gene deliveries to reprogram mouse embryonic fibroblasts into neurons. In this study, we reprogram mouse embryonic fibroblasts by delivering the BAM factor to the cells with a non-viral gene delivery, with a polyplex delivery, with only one dose of polyplex delivery. So after around 2 weeks, we observe a higher percentage of cells expressing both TJ1 as well as MAP2, with the highest expression on a hierarchical pattern, which means that we have 250 nanometers gratings patterns in parallel on top of a 2 microns gratings. On the 2 microns grating and the 250 nanometers gratings, we also have an increased TJ1 expression and MAP2 expression. So again, TJ1 is a marker for a pan-neuronal marker, and MAP2, the myosin-associated protein 2, is a marker for neuronal maturation. We also measure the electrophysiology activity or functions of the neurons, and we find that we could detect cells with single action potential and multiple action potentials when the cells are derived on the gratings pattern, but we couldn't detect any electrophysiological activity in the cells on unpatterned substrate. So this also has give us supporting evidence that topography substrate, especially grating substrate, could enhance the derivation of neurons or neuronal differentiation. However, what is the mechanism behind it? So we hypothesized that nanotopography could modulate focal adhesion arrangement, which will lead to a rearrangement or change in the cytoskeleton contractility, and it could, as a result, change the cellular function. We start looking into the problem by using human mesenchymal stem cell, specifically the neuronal differentiation of human mesenchymal stem cell as a model cell system to investigate what is the mechanism behind topography-induced differentiation. As during the differentiation, we observed a high expression of phosphorylated focal adhesion kinase, we would like to apply a small molecule inhibitor, the PF573228, to specifically inhibit the phosphorylation of the tyrosine 397 site of focal adhesion kinase. Using this small molecule inhibitor, we can inhibit the phosphorylation of the tyrosine 397 site of FAK, and we find that the neuronal marker that usually is upregulated by the topography-induced differentiation is abolished when we inhibit the phosphorylation at the tyrosine 397 site. To further show the rules of focal adhesion kinase played in the topography-induced differentiation, we also use sRNA to lock down the FAK expression in the human mesenchymal stem cell. Indeed, we find that with the knockdown of the focal adhesion kinase, we also observe a significant reduction in the neuronal marker expressions that we usually will see in the topography-induced differentiation. In a collaboration with our collaborator, Professor Lou Roma, in Johns Hopkins University, we use SuperFAC, which is an engineered mutant inducing an increased autocatalytic phosphorylation of FAK. Using SuperFAC, we find that it actually increased the sensitivity of the human mesenchymal stem cells to topography sensing, in which the topography-induced neuronal marker expression is significantly higher when it's exposed to the topography. So it further shows the significant involvement of focal adhesion kinase in the topography sensing. Another questions we would like to ask is what is the role of the force sensing in terms of the force-mediated signal by the cytoskeleton contractility. So in this experiment, we also applied two other small molecule inhibitor, the Y compound, which is the ROCK inhibitor inhibiting the Rho kinase effector, as well as bipistantin, which is an inhibitor for non-muscle myosins 2. When we apply these two inhibitors to inhibit the cytoskeleton contractility, we see the neuronal marker that usually expressed in the cells undergoing topography-induced differentiation. Those neuronal markers expression is being abolished, which shows that the topography-induced differentiation does require cytoskeleton contractility. We would also like to investigate the involvement of cytoskeleton contractility in the embryonic stem cell differentiation or nanotopography. My other student, Solila, has taken this work and she investigates the role of ectomyosin's contractility during the early phase of topography-mediated neuronal differentiation, and she looked at the expression in day 1, day 4, and day 7 after the neuronal differentiation has started. So in pluripotent stem cells, in embryonic stem cells, when they are undifferentiated, they have very low expression of stress fiber or cytoskeleton contractility. So both F-actin expression as well as the phosphorylated myosin light chains expression is quite low. During the differentiation process, the formation of stress fiber as well as the phosphorylated myosin light chains expression will increase as the cell starts to differentiate. So during this process, we find that the cells culture on the 250 meter nano gratings will have stress fiber appeared as early as day 1. So the cytoskeleton contractility developed much earlier on the nano gratings. Very interestingly, when we applied the inhibitor, the Y compounds to the ROCK inhibitor, we actually observed that the ROCK inhibitor, with the presence of ROCK inhibitor, we have a higher expression of TJ1 in the human embryonic stem cells concentrating on the nanogratings. And in this case, it means that when the ectomyosin contractility is decreased, we actually have a higher expression of the neuronal marker. Meanwhile, in the presence of piperacillin, when we inhibit the ectomyosin's contractility, it actually leads to a reduction in MAP2 expression. So these two data seems to be contradicting, but if we think about it, it is actually agreeing with what other researcher has found in the literature as well. In the literature, other researcher has found that using a soft substrate will help to enhance and induce neuronal differentiation. And in the soft substrate, the cells will develop a lower ectomyosin's contractility in response to the softer environment, which is similar to when we apply a low dose of Y compounds to lower the ectomyosin's contractility. And in response, the combination of that ROCK inhibitor with the nanogratings, we observe a higher expression of TJ1. However, as what we have observed in our human mesenchymal stem cell study, as the ectomyosin's contractility is needed for the differentiation, for the topography-mediated differentiation, if we inhibit the ectomyosin's contractility by piperacillin, we will not be able to observe the maturation of the neurons and the expression of MAP2. So when we are talking about the cytoskeleton contractility, we would like to quantitatively examine the ectomyosin's contractility under traction force of the cells cultured on the gratings. In collaboration with our collaborator, Dr. King-Wei Chim, in Bioinformatics Institute in Singapore, we have developed this 3D traction stress measurement system on 2 microns gratings. So first, we observe that the cells on these micro gratings exert a significantly higher force that is in the parallel directions to the gratings. Another interesting observation that we have is that the cells actually exert a higher traction force on each of the focal adhesion. So, but 2 slides ago, I mentioned that the low contractility of a lower contractility will help to enhance neuronal differentiation. Indeed, this is also what we observed. Because, or what we speculate, because when the cells are culturing on a flat or unpatterned substrate, we see many more focal adhesion. But on the grating substrate, we observe fewer number of focal adhesion. So although the traction force per each focal adhesion is increased, when we investigate the overall contractility, we actually find the overall contractility is lower in the cells on the gratings, which also agree with our speculations that the grating substrate will lead the cells to have a lower contractility, which also is one of the reasons why it could contribute to the enhancement in the neuronal differentiation. We also hypothesize that the change in the cytoskeleton may also affect the nucleus, the gene regulation in the nucleus. So we also look into the nucleus morphology at the early stage of differentiation. So on the cells cultured on the nano and micro gratings, we find that the nucleus also significantly elongated on the gratings substrate, and the nucleus is also aligned along the gratings direction. We also observe that there's an increased X3K9ME3 expression in the in the early phase of the neuronal differentiation when we culture the embryonic stem cells on 250 meter nanos grating. So in summary, we speculate how topography influences cells differentiation or cell behavior through mechanism as followed. Topography signals are transduced through focal adhesion kinase signaling. And topography-induced differentiation is mediated by ectomyosin's contractility. And both this ectomyosin's contractility as well as the focal adhesion signaling is important in topography sensing. When we decrease ectomyosin's contractility of the cells, it could enhance the neuronal lineage commitment, so there's more TUJ1 positive cells. However, if we inhibit the ectomyosin's contractility, it could also inhibit the neuronal maturation. So MAP2 expressions, the maturation of the neurons actually depends on ectomyosin's contractility. With using a 3D traction force measurement, we find that cells on gratings exert anisotropic traction stress on the gratings with overall lower contractilities when the cells are cultured on grating substrate. And we also find that the force transduced to the nucleus will lead to a change in nuclear shape and morphology, and it could also affect the gene regulations, leading to subsequent change in cell behavior as well as changing the protein expression. So with that, I hope that I have convinced you that it's important to consider topography or the surface structure of a material, and I'm also able to convince you that topography could significantly affect cell behavior and such as stem cell differentiation. With that, I would like to thank my student again, and also my current student and also my former student that has contributed to the work, our collaborator funding source, and thank you very much for your attention.